All right, good morning, everyone. What we're going to be looking at today is I'm going to turn myself a little bit. There we go. What we're going to be talking about today is diffusion. If you remember, diffusion is particles moving from an area of high concentration to an area of low concentration. This is how cells are going to move particles in and out of, okay? So what we're going to be looking at today is how can diffusion be observed? How do we actually see this happening? Well, in Google Classroom, eventually you'll have access to the sheet. And the sheet is what we're going to do is look at the diffusion of a substance across the semi-permeable membrane. How do you create that? Well, what we're going to be using today is iodine. Iodine comes in a nice bottle like this. When it's poured into a cup, you can see it kind of looks like that. It looks nice and dark. Um, this is known as an indicator for starch. Well, what does that mean, starch? Well, the starch that we're going to use today is your average, just your nice cornstarch that you probably have in your cupboard. So with that, as I'm going to throw some water into a cup, okay, I have some cornstarch in here, and I'm just going to mix it up. Cornstarch and water, that's it, okay? Now, what does it mean to be an indicator? An indicator says that it'll change color in the presence of it. So if I'm looking at this, I'm going to put 2 drops in. 1, 2. And now what I'm going to do is, if you watch this, and as I change it, what happens to the color? It's changing to this purple color. That means that it's indicating that there's starch. I'll put a couple more drops in. It's indicating that there is starch in here. And you can see how that went from milky white to just a purplish color. So that's what an indicator is. So iodine is an indicator for starch, okay? And how are we going to show this? Well, we're going to take a plastic baggie that looks just like this, and your run-of-the-mill plastic bags from the store, okay? And what I'm going to do is I'm going to take a couple of scoops of cornstarch and put it in there. And here we go. I'm going to take a couple of scoops of cornstarch. I'm going to put it in. Pretty simple. There we go. Now I'm going to go put water in. I'll be right back. All right. Now, sorry, but I put the water in, and here's the water. And why don't I just make myself full screen? All right. Now, here's my water. I'm just going to mix that up, and that's just cornstarch and water. So now, what I'm going to do is I'm going to tie this. Okay? And, of course, if you guys were in class, you'd be doing this lab. But, next best thing, huh? Watch Mr. Teskey do it, I guess. Good or bad, whichever way you look at that. All right. So, I'm just going to tie it in a knot. And there's my cornstarch and water. The second part of this is I'm going to take another cup, which let me dump this one. I'm going to take another cup and just put water in it, and then I'm going to take iodine and put it in there. Okay? And all I'm doing is putting the drops of iodine. You can even see. I'm just putting, here, let's go like that. There you go. And I'm putting the iodine in, and I'm just going to take a couple of squeezes here of iodine just so it shows it. Now, in this lab now, I'm going to then, here's my iodine. Here's my cornstarch. And I'm just going to dunk it in there like that. I'm going to leave this for 15 minutes. At the end of 15 minutes, we're going to take it out. Now, what I want you to think of is, when you're looking at the lab, is look at how or what is going to happen. If we're saying this bag, the bag is representing a cell, and this is our selectively or semi-permeable membrane. Now, if the cornstarch is able to diffuse out of the bag and into the iodine, what'll happen? Or will it be the other way around and the corn or the iodine will go in to the bag? How would we know? That's where the indicator comes in. So now what's going to happen is you are going to look at the sheet. I'm going to minimize this. And on the sheet here, while we're waiting, you're going to go through and fill out these questions. And when we look at what's in the bag, you can explain that of which constant, or like, for example, which is more concentrated in the starch? Is the highest concentration of starch in the bag or in the beaker? Well, the bag, because it's all starch and there's none in the beaker right now or in the cup. All right, and then it's going to ask some predictions, and then the starting color. So here's the starting color of the baggie, which is white, and here's the starting color of the iodine, which is kind of a, what do you call that? An orangish, purplish type color. Okay, so that's the two differences between those. And what I'm going to do now is I'm going to stop this video and I'll upload the video of what happens 15 minutes later.